Okay, so let's see here. We left off here, right? Last time? Okay, so now let's get into specifics. So as we get into specifics, I want to reiterate something that I always tell you guys, but this time I really truly mean it. So if you ever were to take my advice, now would be the time. And that is that, as you'll see, we're going to get into the nitty-gritty. Very simplified, but still a lot going on for these cell signaling pathways. So we're leaving out a lot of players. Keep that in mind. So in your hate of me, just think that you could have hated me more. So the book simplifies, I simplify too, to make them as straightforward as possible. But there's still a lot of players involved, as you'll see with these signaling pathways. One thing activates the next, and then that activates the next, and then that activates the next, and then there's some sort of specific cell response in specific cell types. So what does that mean? That means a lot of memorization for chapter 16 of these sort of minute little things that are happening in a particular sequence. That just means that you should take a little bit, you should start early, because the earlier you start, the better the memorization of these things will be. Because it is a lot. There's no way around it. These pathways, there's a lot of players involved. Okay, so we're going to get into talking about specific signals that lead to particular results in certain cell types. Starting with acetylcholine. So acetylcholine, we've already talked about acetylcholine as a signal, and we're going to continue to do so in several different examples. So here's what I'm going to do as a sort of shorthand. So I suggest perhaps doing this as well as you're going along and studying. This is a good shorthand, and just see if you have your pathways down. So in heart, I should start with the cell type. So in heart pacemaker cells, acetylcholine does what? So acetylcholine is a signal, and I'm just going to draw a series of arrows if it induces the next thing to occur, or this upside down T if it's blocking that next thing from occurring. So what's going on in heart pacemaker cells? Okay, what are heart pacemaker cells to start? Heart pacemaker cells are the cells in your heart that are regulating how fast or slow your heart rate is. So when they are electrically excitable cells, like neurons are electrically excitable cells and muscle cells are electrically excitable cells. But what they're doing when they're active is allowing the parts of the heart to pump. We're in a dress today. I don't know where to put this thing. Okay, so how do they do so? They do so in response to a signal. So the signal is acetylcholine, or they change their levels based on responses to signals. So acetylcholine is the signal. The receptor is called the acetylcholine receptor, although we know that there are different types, right? So this is a particular acetylcholine receptor that is a GPCR. So GPCR becomes activated, activates a G protein. This particular G protein, remember there are different GPCRs that are activating different G proteins. This particular G protein is called G subscript I. What is GI doing once it becomes active? And it's actually so it does what I just told you, right? It splits off into two. And it's actually the beta-gamma complex, so we can kind of maybe make a note of that. It's the beta-gamma complex that's involved in this process, which is there is a potassium channel at the cell surface of these pacemaker cells that are ligand-gated and open up in response to beta-gamma. So beta-gamma activated, splits up, diffuses laterally through the plasma membrane, finds the potassium channel, and allows it to open up. What happens when the potassium, and therefore potassium, can exit the cell? What happens in electrically excitable cells when potassium leaves the cell? What does that do to the membrane potential? Hyperpolarizes that membrane, makes it more negative, makes it much more difficult for these electrically excitable cells to start an action potential. So what we get is hyperpolarization, or the real result is a decreased firing or a decreased heart rate. So even though in response to acetylcholine we have a channel that opens up, this isn't the same thing as these ion channel coupling receptors. So it is in the end going to influence an ion channel, but it does so not directly. So it's not directly coupled to an ion channel like the acetylcholine receptor that we talked about at the neuromuscular junction is. So it's coupled to a G protein, which will then activate the ion channel. So there's a distinction there. This is a GPCR. And GI we can remember. I remember that it opens up an ion channel because of ion, but the I actually stands for inhibitory, because in this case acetylcholine is an inhibitory signal. So it's going to decrease in the end the possibility of an action potential. Okay, so that's one example. So if we go back to this slide, acetylcholine is doing lots of things. It's skeletal muscle, it's excitatory, it's allowing for contraction, salivary gland cells. We talked about how, we didn't talk about the pathway, but we talked about how it leads to secretion of saliva via vesicle fusion. Now we know how it allows heart pacemaker cells to decrease the rate of firing, and therefore lower the heart rate. We also talked about a fourth thing acetylcholine does. Does anyone remember? We talked about that on Monday. It wasn't that long ago. And surely nothing's happened since then. It's real fresh in your memories. Does anybody remember what acetylcholine does, that fourth thing? What? Opens blood vessels, yes. But let's talk cell biology, the exact cell response. The one, acetylcholine does what to these cells and allows them to do what? The overall physiological response is an opening of blood vessels, but what's actually happening? Which cells? And what's the result? The endothelial cells. So acetylcholine does that fourth thing. In endothelial cells, what's the result? Release of nitric oxide. So that's, which will eventually lead to opening up those blood vessels. So that's another thing acetylcholine does. Number 4. Okay, so what a lot of these, again, there are different G proteins that become activated. We just saw GI. A lot of these G proteins, how they function is that when they become active, they are going to, again, diffuse laterally and then bind to their binding partners and activate them. So a lot of times, in a lot of cases in these different examples, what they activate is either adenylyl cyclase, which is an enzyme, or phospholipase C, which is another enzyme. A lot of these pathways, the way they work, is G protein activating these two enzymes. So what are these two enzymes doing? These enzymes, when they become active, make secondary messenger molecules. So small molecules, an example of which I already gave you, which was cGMP. But here's how they work. So once the enzyme is active, it responds to G proteins activating them. If substrate is available, then they're just going to take substrate and convert it into product. And that product is a secondary messenger molecule. So as soon as it becomes becomes active, take substrate, change it, make product. Take substrate, change it, make product. Take substrate, change it, make product. And so product, the secondary messenger molecule, is going to keep increasing in concentration and diffuse throughout the cytosol and pass that message along. So the amplification of the message a lot of times happens at this step when the enzyme that becomes activated makes all of these secondary messenger molecules. So then what happens to turn the message off? We have to turn the G protein off. We know how that happens, right? Hydrolysis of GTP. And that's going to turn the enzyme off. But then we also have to get rid of all of these secondary messengers. So there has to be something to get rid of the secondary messenger in order for the cell to return back to its previous state. Okay, so the two most common target enzymes, as I said, adenylyl cyclase and phospholipase C. So let's first talk about adenylyl cyclase. So GPCR, once active, activates a G protein. GS is the G protein that S for stimulates, or stimulatory, that activates or stimulates the activity of adenylyl cyclase, also commonly known as adenylate cyclase, same enzyme. So adenylyl cyclase is an enzyme that catalyzes this reaction right here. It takes ATP, the substrate, and makes cyclic AMP in a very similar, analogous way to what guanylyl cyclase does to make cGMP. So here's ATP, removal of these two phosphates, and then cyclizing that third phosphate onto the ribose ring makes that cyclic AMP. Now, ATP, not a messenger molecule, cyclic AMP, secondary messenger molecule. So the more of it gets made, there's lots of ATP available in the cell, the more of it gets made by adenylyl cyclase, it's going to diffuse throughout the cytosol and be a messenger molecule. Then what happens? At some point we want to return to the off state. So again, all of that cyclic AMP, we have to get rid of it. We don't want it to stick around and keep the message on. So cyclic AMP phosphodiesterase is the enzyme that gets rid of cyclic AMP. It hydrolyzes this magenta bond right here, phosphodiester bond, and makes cyclic AMP into regular old AMP, which is not the on state for the cell. So what if we were to inhibit cyclic AMP phosphodiesterase with a chemical inhibitor? We would keep the cell in the on state longer. And such a chemical inhibitor exists, and I'm very much addicted to it. It is caffeine. So caffeine inhibits cyclic AMP phosphodiesterase, and for certain cells that means remaining in the stimulated state a little longer. Very much addicted to caffeine. It's a problem. Okay, so we don't talk about serotonin in terms of cell signaling pathways in this class, but serotonin is an important neurotransmitter signal. And in response to serotonin, we get this occurring, which, whoops, I stopped there, but bless you, that is a very cute sneeze. Cyclic AMP is made. So in response to serotonin, so serotonin is released by certain neurons and is received by other neurons, and then they respond in this way. And so this is just to show you, this is an image just to show you how quickly that can happen, and cyclic AMP starts diffusing throughout the cell. So this is a neuronal cell. It has within it a fluorescent protein that, when bound to cyclic AMP, fluoresces. So it can be utilized as a cyclic AMP marker. And so this is kind of a heat map. You know, like heat map, where red indicates lots of heat. Same sort of thing, but it's a heat map of cyclic AMP. So that red means that there are high levels of cyclic AMP, yellow less so, and then green, and then finally blue means basically no cyclic AMP. So the cell starts off with basically no cyclic AMP. And if you add serotonin very quickly, within 20 seconds, you can see that we have these really high levels of cyclic AMP, and then it's starting to diffuse so that there are lower levels, but it's starting to diffuse inward into the cell. So within 20 seconds, cyclic AMP levels increase by about, I think this is like 100-fold or more, and then, not 100-fold, I don't know, I can't remember the numbers, but 1,000-fold, 100, yeah, something big. And then it starts diffusing out into the cell. Again, within mere seconds. So this occurs, this, and then adenylyl cyclase made a lot of this, and then it started diffusing all within just a few seconds. And then, of course, not shown here, but adenylyl cyclase is going to remove that cyclic AMP from the cytosol. Okay, well, what's cyclic AMP doing? It's doing a lot of things, but one of the major things that it does is bind to and activate a protein called protein kinase A, or pKa for short. Here's pKa. pKa is a protein made up of several subunits. Two of those are regulatory subunits, and then the other two are catalytic subunits. So when the protein is all together in its 4 subunit structure as a tetramer, it is in its off state. Cyclic AMP binding to pKa here at these regulatory sites allows for pKa to become active. So in its active state, kind of like that G protein, the trimeric G protein, it splits off. So cyclic AMP allows for these inhibitory regulatory subunits to come off of the catalytic subunit. Now, what are those catalytic subunits doing? What type of catalysis, what type of reaction are they allowing to occur? It's a kinase, protein kinase A. Yeah. These go on to phosphorylate and activate other proteins. So once pKa becomes active, it phosphorylates and activates other proteins. So, and here we have another, maybe a better image from the previous one of how that works. So again, at serines, thurines, and tyrosines that have available hydroxyl groups as their side chains, the kinase's job is to covalently add a phosphate to those hydroxyl groups. And then phosphatases remove the phosphates. So typically protein kinases, when phosphorylating proteins, activate them, and phosphatase is when removing those phosphates, deactivate them. But that's not always the case, but that's typically the rule. Okay, let's look at a pathway. Let's go back to skeletal muscle cells. What's happening in skeletal muscle cells? A lot of things, right? They can be contracting and relaxing. They're also receiving other messages that are not just contract and relax. So what are those other messages? Adrenaline. Is the signal molecule, the extracellular signal molecule. What's adrenaline? I'm sure you guys have all heard of adrenaline, right? What's adrenaline? It's epinephrine, yeah, that's another name for it. It's up there, I think, yeah. What's epinephrine / adrenaline? Have you all heard of it? Fight or flight, that's the one, right? The fight or flight response releases adrenaline into your bloodstream. A lot of cells respond to adrenaline, not just skeletal muscle cells, but let's get specific about what happens in skeletal muscle cells. So fight or flight, what does that mean? That means I'm scared. I just had adrenaline this morning because a guy cut me off. And I could feel what happened. It was an immediate response because I was about to crash. So, was that fight or flight? I don't know. I guess it was breaking. I don't know which one. But adrenaline released in times of these stresses, like I'm about to crash, like a bear is chasing me is the evolutionary example, or a not a dinosaur, no. A bear, I don't know, woolly mammoth, that we were around, right?
Yeah.
Okay. So yeah, it's your fight or flight. It allows you to respond quickly to these cases of stress. Now, adrenaline, when you're giving a presentation or when you're taking an exam, is what are our stresses these days, but still called fight or flight. We're running away. Let's say we're running away from a bear. Okay, so what needs to happen? All sorts of things. Let's talk about what happens in your skeletal muscle cells. Adrenaline reaches the adrenergic receptor. The adrenaline receptor is a better name for it, which is a GPCR, which activates GS. What does GS do? Activate adenylyl cyclase. What does adenylyl cyclase do? Make cyclic AMP. So as you're doing these, you can also do shorthands for, I don't know, make, if you want to, or binds to, or phosphorylates. As shorthand, so binds to and activates. So how does it activate, or how does it go on to the next step is another thing that you can add to these. So cyclic AMP binds to pKa. pKa is a kinase, and so it binds to and phosphorylates this protein right here called phosphorylase kinase. So I guess we can do a P there. Phosphorylase kinase. Phosphorylase kinase is in the name. What is it doing? It's a kinase, so it's going to bind to and phosphorylate another protein. And a lot of times these kinases, not always, pKa isn't the case, but sometimes these kinases are named for what they phosphorylate. So phosphorylase kinase phosphorylates a protein called glycogen phosphorylase. What's glycogen phosphorylase doing? It is an enzyme whose job is to break down glycogen. So, upon becoming active, upon phosphorylation, glycogen phosphorylase takes glycogen. Do we remember what glycogen is? Storage of glucose molecules. So it is a glucose storage molecule that is not found really in all of your cells, but muscle cells have stores of glycogen. And this is a sort of fast way. So as soon as glycogen phosphorylase is activated, it'll break those glycogen, glycoside bonds between the glucoses to create available glucose, which the skeletal muscle cell can use to break down in glycolysis and create an ATP, which we already know the skeletal muscle cell needs a lot of to restore calcium levels back to where they were with that calcium pump, for example. And we're going to talk about other ways in which it needs ATP in order to contract. And why do we need to contract? If we just bring it back a little bit to the physiology, is because a bear is chasing us. So our muscles need energy in the form of ATP so they can break down those glucose molecules. So, glycogen breakdown is a result of that. What else is happening? You don't need to worry too much about this because we're talking specifics about different specific cells, but all kinds of things, right? So your heart rate increases. That's also going to bring more blood and more oxygen and perhaps more glucose to your muscle cells. The pupils dilate, I think. That's another response to adrenaline. I don't know, maybe because you're running away in the dark from the bear, I don't know. But, yeah. So, Here's one cellular response in response to adrenaline or epinephrine. Now pKa is a central player in lots of different cells. So the activation of pKa via these cell signaling pathways is how a lot of cells work to do different things for the cell. So pKa is what we call a promiscuous protein. And that's the actual term for proteins that do what? That bind to that have a lot of different binding partners. We call those promiscuous proteins. pKa is promiscuous, and then it has a lot of different binding partners. So if the binding partner is there, the protein is there in the cell, then it's going to bind to it, phosphorylate it, and activate it. So depending on the cell, what pKa activates really depends on which proteins are available for it to activate. In skeletal muscle cells, it's going to be phosphorylase kinase is one of them. In other cells, so remember adrenaline is doing other things. In other cells, pKa and a response to adrenaline is going to be a slower thing. It's going to physiologically and cellularly going to be a slower response. So in neuronal cells, for example, it is thought that pKa, well, it's not thought, it is seen, but then why the next thing happens is a little bit more confusing and not certain. But it is seen that pKa will activate by phosphorylation different transcription factors that's going to lead to transcription of certain genes. So again, this is a slower process, but it is believed that, let's say, in neuronal cells, this leads to things like proteins that are made that lead to long-term memory associations. So this is why, partly one of the reasons, not fully understood yet, but why you remember, if a bear chased you, you probably remember that for the rest of your life, as opposed to just what you had for dinner last night. So that's where it's a longer process through transcription of certain genes, but that is involved in long-term memory. Whereas this response in your skeletal muscle cells is fairly immediate. As soon as GPCR is activated, it activates the next thing and the next thing and the next thing. All the proteins are there. All they need to do is become phosphorylated, which occurs really quickly, and we get glycogen breakdown happening fairly quickly within those cells. Because it wouldn't give the muscle cells any good if an hour later now the glucose is coming in, and now you can run away from the bear. You're already I was going to make a joke. What's that movie with Leonardo DiCaprio and the bear? I forgot the name of the movie. Okay. Yeah, another thing that happens is, say in endocrine cells, adrenaline signals those cells to secrete another signal. So it's kind of, in all of these, we're focusing on cells and what that immediate result is. But remember that in all of these, the result isn't just in one cell. So that leads to something else happening in the body. Okay, so phospholipase C is the other enzyme that's pretty commonly activated in response to an extracellular signal, and GPCR is becoming activated and activating G proteins. So GPCR activates a G protein. In this case, the G protein is called GQ, and I have no easy way no mnemonic device for how you remember GQ activates phospholipase C, but it does. I did mention this, GS, right? Did I? Let me go back. Yeah, GS. S stands for stimulatory. We talked about that, right? Okay. So the S in this case, stimulatory activates the endothelial cyclase. The GQ in this case, no idea what it stands for, but it's going to activate the enzyme phospholipase C. Phospholipase C. this enzyme right here. So what's phospholipase C doing? Phospholipase C is an enzyme that also creates secondary messenger molecules. So the substrates need to be readily available in the cell, and once active, it's going to create a secondary messenger. And actually, it creates two of them. So how does it do that? The substrate is right here. The substrate is the inositol phospholipid. Do you remember the inositol phospholipid from chapter 11? This is the phospholipid with the inositol head group. And I told you inositol is special in that it can be phosphorylated and act in cell signaling. So, and that's shown here. Here's the inositol head group, which is a sugar, and it has been phosphorylated 3 times, so that we have 1, 4, 5. Those are the places within the sugar where carbons in the sugar that have been phosphorylated. So this is a phospholipid. Phospholipase C cleaves right there, basically removes the head group from the lipid. So that's what phospholipase C does. So here's the head group. It has been hydrolyzed from the lipid. So now it's its own soluble, cytosolic soluble thing called inositol 1,4,5-triphosphate, or IP3 for short. So the P3 standing for the 3 phosphates that are attached to the inositol. Now, the lipid that remains, it's lost its head group, and so now it's a simple diacyl for the 2 fatty acids, glycerol. So diacylglycerol is at the plasma membrane, and both of these, so again, phospholipase C, if active, it's just going to cleave a bunch of these IP3s from the diacylglycerol, creating an abundance of these secondary messengers. Diacylglycerol is the other one. This one's soluble, so it can diffuse throughout the cytosol. This one's lipid soluble, so it can diffuse laterally on the leaflet of the plasma membrane, inner leaflet of the plasma membrane. And both of those go on to pass the message along. So what happens? IP3 diffuses through the cytosol, finds this calcium channel at the membrane of the ER. Now, remember, no, don't remember. I don't know if I mentioned this, but the ER does a lot of things we talked about, right? Protein synthesis, for secretion, lipid synthesis, all of that is true. The ER is also an organelle that stores calcium inside of its lumen. In that way, it kind of acts like the sarcoplasmic reticulum. So calcium is found inside the ER lumen. There's some calcium pump that pumps the calcium in. We didn't talk about that, but there is. And then IP3 opens up this calcium channel, allowing calcium to enter, to flow into the cytosol. Now, diacylglycerol, which can diffuse through the membrane, it has a job too. It's also a secondary messenger, but it's just at the membrane. So it binds to and activates, it actually brings to the membrane this protein called PKC. What do you think PKC stands for? We have to guess. Protein kinase C. Yeah, it's not the most original, but that's the name, protein kinase C. So now we have calcium in the cytosol, which, surprisingly enough, also helps to activate PKC. So both of those things are necessary. Binding to the diacylglycerol and calcium binding to PKC. Both of those things are necessary to activate PKC. And what do you think PKC is going to do? Phosphorylate and activate something else. So that's how that works. And I'm just pointing out here that both the alpha and the beta gamma, both subunits are necessary to activate phospholipase C. So this is a convoluted, but in the end it works together to have some sort of result for the cell. So what is that result? Do we have time to talk about it? No, but I do have time to show you this video. I think, and then we can go. So calcium signaling is something we've already seen. The cell utilizes calcium signaling a lot. Here's an example. This happens right after sperm reaches egg.
The sperm cell fuses with the seed of the egg cell.
That's a fake sperm.
In these experiments, calcium concentrations are visualized and measured with a fluorescent dye that becomes increasingly brighter the more calcium.
So same thing, it's a heat map of calcium levels.
Brightness is then translated into a color signal.
And see how quickly the calcium goes through the egg cell. That's it.
Red and high.
All right. See you all on Friday.